Hello, in this video we will learn about the RNA splicing mechanism. So we will take the example mRNA, messenger RNA. So let's begin to understand. This is the cell content DNA as a genetic material will be transcribed into the mRNA, messenger RNA. This messenger RNA is the pre-messenger RNA contain the intervention of the intron and exon. Exon is the useful file. The intron is it is not useful and should be removed. So how it remove, that is known as the RNA splicing mechanism. Anyhow, this mRNA will go into the cytoplasm from the nucleus, and this mature mRNA contain poly A tail and the 5' cap, and it is contain exon only, so it is able to translate the protein when ribosome will bind, is known as the polysome formation. So anyhow, the basically intron is the unrequired short sequences within the mRNA will be removed to exon left. Anyhow, the spliceosome is used for splicing. It contains catalase enzyme used for the catalyzing. But in this way, we will not go into detail but understand the major concept about the RNA splicing mechanism. Anyhow, this is the exon and intron containing. Inside is the intron, while the peripheral part of the sequence is the exon 1 and exon 2. And this is the 5' splicing junction within the intron and 3' splice junction, and the branch point adenosine are present here. Anyhow, this is a 5' and 3' and 3' and 5' and to the mature exon 1 and 2, and the here intron will be removed as a lariate. This lariate contains 3' here, and 5' will be added with the adenosine. So the splicing involves two successive strands esterification reaction in which one phosphodiester linkage is replaced by another. In the first reaction, the 2-hydroxyl group of the branch point adenosine attacks the phosphate group in the sugar phosphate at the 5' splice junction. This cleaves the sugar phosphate backbone of the RNA strand. Creating loop in the RNA molecules is known as the lariate. So remember, let's going to understand with the detail. This was the theoretical explanation. You will not understand completely, but let's begin to understand via a step. So this is the sugar phosphate backbone of the RNA strand. Creating loop in the RNA molecule is known as a lariate here. So let's begin to understand step 1 of the RNA splicing mechanism. How it is possible? Let's here via a diagram. To briefly understand, this is the exon 1 and exon 2. Exon 1 is initiated from the 5', while the exon 2 is initiate and in the 3', and this is the intron. This intron is a green color is showing in the diagram. I will color it. So the branch point adenosine are present here, and this is the adenosine. Remember, in this way here is the exon 1 and exon 2, and this is attached via a phosphate group, phosphodiester bond. So in this way here you can see this the 2-hydroxyl and are present with the sugar, and in this way the water will be attack here, and in this way the spliceosome is using for it, and in this way here is the exon 1 end of the exon 1 will donate the hydrogen to the water to produce the 3 water proton. So the 2' hydroxyl of the branch point adenosine attack the phosphate at the 5' splice junction, releasing exon one with a free 3 hydroxyl group. So one with 3 hydroxyl group free. So anyhow, let's begin to understand the 3 prime end of exon 1, which was released in step 1, and attack the phosphate at the 3 prime splice junction, and in this the attacking to each other and connecting exon 1 and 2, which releasing the intron. How it is possible? Let's begin to understand. This is the 3 prime side contain a hydroxyl group, and in this way the water will receive the proton to form a hydronium ion proton. And while here you can see this is the adenosine basically here, and in this way intron will be removed, and in this way the adenosine combine together to each other and in the form intron. While this is the intron will remove in this way, removing through exon 2 with the phosphate group. In this way the proton will be donate to the water, and in this way here and here you can understand this is the reaction will occur, and in this way the intron will be removed as a laureate. And let's begin to understand with the written the 3 prime end of exon 1, which was released in step 1, attack the phosphate at the 3 prime splice junction connecting exon 1 and 2 while releasing the intron. So after this, the releasing the intron, basically the 3 prime attack to the 5 prime side of the phosphate group to exon 1 and exon 2 to each other is known as the trans-esterification. This trans-esterification will lead to products. The products consist of exon and 2 1 and 2 exon, which are now connected in a continuous RNA strand, and the intron byproduct, which exists as a looped laureate structure. Anyhow, intron is here, and in this way the loop-like structure will be formed. Adenosine attached together to form a loop-like structure, while the exon 1 and 2 will be attached via a trans-esterification, and in this way the exon 1 and exon 2 combine together, and in this way the whole long mRNA will be formed. That mRNA is the mature, is able to synthesize protein after the binding of the ribosome. That is known as translation after the transcription. Thanks for watching. If you like this video, please make sure to subscribe, like and share. And this video was about the RNA splicing mechanism, and we get exemplifying the mRNA. Thanks for watching. If you like this video, please make sure to subscribe, like and share, and see you in the next video. Bye.